Good evening, students. Today we will see the reasoning of questions. Why red blood cell is called so? Yes. So the red blood cell is called as red blood cell because of the presence of the hemoglobin pigment, which is a red color pigment. Second question, how RBC of large diameter pass through the smaller capillaries? Yes. The RBC, though it is of large diameter, it is biconcave shape, and this biconcave shapes makes the RBC to get folded and pass through the smaller capillaries. Third question, why fetal hypoxia is more common? Yes. So fetal hypoxia is more common because of the hemoglobin which contains the HbF hemoglobin, and this HbF hemoglobin has a more affinity for oxygen, so the delivery of oxygen will be less. Fourth question, why sickling occurs in hemoglobin S? Yes. So due to the point mutation in the beta chain, you have hemoglobin S. So the glutamic acid is replaced by a valine acid, amino acid, and this causes polymerization of the hemoglobin at low oxygen levels. And that is why it causes a sickle-shaped RBC. RBC with hemoglobin S will be of sickle shape. Fifth question, why hemolysis occurs in sickle cell anemia? Due to the rigid shape, while passing through the capillary, breakdown occurs because of the sickle shape. So the normal RBC shape is lost. That's why hemolysis occurs. Thank you. So good evening, students. We will continue the blood. Okay. So why RBC dies out in 120 days? This is the next reason question. Because of lack of organelles. The RBC does not have nucleus, mitochondria, and since it is not having nucleus, it is not able to produce new proteins and new enzymes. Okay, so since it is lacking mitochondria, it depends on anaerobic glycolysis, so it is not able to get new enzymes, so it dies out in 120 days. Why erythrocytes sediment when they are allowed to stand in a test tube? It is due to the lack of it is due to the property of Rayleigh formation. What is Rayleigh formation? The RBC will pile up one above the other, and this formation is helped by the fibrinogen. So it acts like a glue that makes the RBC to pile up. Why helper T cell is called so? Yes, it is called helper because it helps the B lymphocytes or B cells to get activated to plasma cells. And once the B cells converted into plasma cells, that produces antibodies. Next question. How the T cell identify the antigens presented by the antigen presenting cell? Yes. So it is identified by the major histocompatibility complex, MHC complex, which acts as a marker which is present on the antigen presenting cells. So so here the CD4 helper T cell will recognize the antigen with MSC2 marker on the antigen presenting cell, whereas CD8 T cell will recognize the antigen with MHC1 complex on the antigen presenting cell. So antigen presenting cell with MHC2 complex with the antigen will be recognized by CD4, and MHC1 complex marker antigen presenting cell will be recognized by CD8 T cells. The next question, how the antigens are attacked by antibodies? Yes. So by precipitation, neutralization, opsonization, and agglutination. Precipitation means the antigen will get agglutinated. It is more of agglutination. It forms a complex with the antibody. Antigen antibody complex will be formed. Neutralization is the toxins produced by the antigens will be neutralized by the antibodies. And opsonization is the antibody will coat the antigen, make it edible, so that the macrophages will engulf the antigen. Okay, the next question, on what basis immunoglobulins are classified? They are based on the heavy chains. We have alpha chain, beta chain, gamma chain, delta chain, epsilon chain. Based on that, it is classified as IgA, alpha chain, IgD, delta chain, IgE, epsilon chain, and uh, so these are the IgG, that is gamma chain, and IgM, that is mu chain. So based on alpha, gamma, um, mu, delta, and epsilon. Why myasthenia gravis is called an autoimmune disease? Yes, here the antibodies are produced against the acetylcholine receptors, and actually this question will be in the muscle. Okay, it was added here, and because the antibodies are produced by the body against this own acetylcholine receptors presented in the neuromuscular junction, it is called autoimmune disease. The next question: why graft rejection occurs after organ transplant? So it is due to the T cells of the recipients, and these T cells will act against the MHC markers. Okay, so MHC matching should be done before the organ transplant, and if the matching is not proper, then the transplant will be rejected. Next question: how platelet help hemostasis? The platelet forms a platelet plaque, which is a temporary plaque formed by the aggregation of the platelet after their activation. So this forms a temporary plaque that arrests the bleeding. Why menstrual blood does not clot? Because of the presence of the substance called fibrinolysins. And this fibrinolysins will lyse and liquefy the clot. How clot lysis occurs? It is occurring due to the presence of the system called fibrinolytic system, and there is a protein called plasmin, which is derived from plasminogen, and this plasmin will lyse the fibrin and clot get lysed. Next question: why the intrinsic and extrinsic pathway of coagulation occurs? The intrinsic pathway, why it is called so? Intrinsic is called so because the activation of the coagulation cascade occurs from exposure of blood to the damaged endothelium inside the vessel. Once they are exposed to the collagen, there will be activation of factor 12, high molecular weight kininogen factors, and subsequently all the factors will get activated. Extrinsic, it is called extrinsic because of the activation of the cascade due to the factor 3, which is the tissue thromboplastin factor released from the damaged tissues. Why hemophilia occurs? Hemophilia occurs due to the deficiency of clotting factors, and classical hemophilia is due to the lack of the clotting factor 8. We have hemophilia A, B, and C. A is due to the deficiency of factor 8. B is due to deficiency of factor 9, and C is due to the deficiency of factor 11. Why thrombocytopenic purpura occurs? Thrombocytopenic purpura occurs. Purpura means purplish spots, and thrombocytopenia means low platelet count. It is due to the blood leak due to the ineffective platelet plug because platelet count is less and blood will be leaking, and due to the leakage of blood, bilirubin, biliverdin will be formed, and bilirubin will be formed that forms the purplish spots. The next question, why RH incompatibility is more significant? Because it causes the disease called erythroblastosis fetalis, hence it is more significant. Why and how cross matching is done? So cross matching is done because to avoid any mismatched blood transfusion or any agglutination reactions. How it is done? The donor RBC is matched with recipient plasma. And if your matching is done, if there is a clumping is there, the blood transfusion will not be done. Okay, so it is done to prevent the agglutination in the blood transfusion. Why RH blood group is named so? The next question. Yes, it is so because it is discovered from a rhesus monkey. So when the rhesus monkeys RBCs are injected into the rabbit serum, they found out there is antibody production, and the same rhesus monkeys antigen when injected in human body, or they produces antibodies, and they found out a factor called RH factor. On what basis the blood groups are classified? Blood groups are classified based on the blood group antigen present on the RBC. If A antigen present, it is A group. If B antigen is present, it is B group. If there is no antigen, it is called O group. If both antigens are present, it is called AB group. The next question, why jaundice occurs in erythroblastosis fetalis? So, yes, it is due to the hemolysis, and because of the hemolysis, bilirubin will be elevated, and that causes the jaundice. The next question, why edema occurs in hypoproteinemia? See, proteins are required for oncotic pressure. So hypoproteinemia means low protein level. So the answer is due to the low protein, there is low oncotic pressure, and that leads to movement of fluids from the blood vessel to outside, and that causes edema. Next question, why anemia occurs in renal failure? So kidney is required for the synthesis of erythropoietin, which is a very important hormone regulating erythropoiesis and RBC production. So renal failure. In renal failure, there will be lack of erythropoietin. That leads to low blood count, low RBC count. The last question, why macroblastic anemia occurs in vitamin B12 deficiency? Yes. So in vitamin B12 deficiency, it is one of the factor vitamin B12 required for the maturation of the RBC. Lack of this B12, it leads to immature RBC, and large size RBC will be there. And because of this large size RBC or megaloblast, there is anemia called megaloblastic anemia occurs. Thank you.